4 billion years ago, Earth was a water world, without life, yet somehow here is when it all began. Somewhere in this time frame, the first signs of life also appeared. There are two theories about how this happened. The first is abiogenesis, life started on Earth. The second is panspermia, life came from somewhere else and was transported to Earth by asteroid. This would be challenging to prove, and it would just kick the can down the road. We also found some puzzle pieces about how life could start in the early Archean, though this is all speculative. First, we should look at what building blocks are present in all life. There are 4 categories: carbohydrates, sugars. These are used as an energy source and for structure. The second are amino acids, which are used to make proteins for enzymes and some complex functions. The third are nucleotides, which are used for information storage but can also be used as enzymes. And the last are lipids, fats. These are also used for energy storage as well as for membranes. For life to form, these should all be present on Earth before there were any life forms to make them. So, is it possible to create all of these without the intervention of life? Well, let's start with the carbohydrates. Vital carbohydrates, like ribose, must be present in the early Archean to support life. There is no method to produce sugars starting from formaldehyde, known as the Formose reaction. Formaldehyde contains the same atoms as carbohydrates, namely hydrogen, carbon, and oxygen. It also contains these in the same proportion, 2 hydrogen atoms for every carbon and every oxygen atom. This reaction is catalyzed with calcium ions. So, in the early Archean, calcium-containing minerals could create carbohydrates from formaldehyde. This has been tested with calcium-containing borate minerals. In a lab, simulating early Earth conditions, this reaction was performed successfully in 2004, generating pentoses, carbohydrates with 5 carbon atoms, the most important of which is ribose. The borate in the minerals also appeared to stabilize, preventing these from falling apart. A more complex class of molecules is the amino acids. There are 20 essential amino acids used by humans, ranging from simple ones like glycine to much more complex amino acids like arginine. Amino acids can form long chains called proteins. While these molecules are more complex, they were also available in the early Archean. In 1952, Stanley Miller and Harold Urey designed an experiment called the Miller-Urey experiment. They simulated the early Archean atmosphere containing methane, ammonia, hydrogen, and water. To approximate frequent lightning, an electric arc was used. The resulting water contained some amino acids. Later experiments added hydrogen sulfide to the mix, and sulfur-containing amino acids such as methionine and cysteine were now also created. While methane, ammonia, and water are quite common on Archean Earth, hydrogen is not common at all. It's so light it escapes the Earth's atmosphere into outer space. However, when a meteor strikes the Earth, the heat from the impact vaporizes the ocean, and the hot steam reacts with the metal in the asteroid, separating water into oxygen and hydrogen. Oxygen is quickly consumed by the ion dissolved in the ocean. The hydrogen stays in the atmosphere until it escapes into space, which takes millions of years, during which the Miller-Urey atmosphere exists on Earth. In addition, during the early Archean, the late heavy bombardment happened. Frequent asteroid impact means that hydrogen was produced this way many times. For the next group of the biomolecules, the nucleotides, there are 5 important ones. These are the building blocks of the DNA. In the early Archean Earth, These molecules could be formed starting from ammonia and methane. Under UV light, these two together can form hydrogen cyanide. While cyanides are mostly known for being very toxic chemically, the carbon-nitrogen bond in this molecule gives a starting point for nitrogen-containing biomolecules, like nucleotides. This hydrogen cyanide can form in the early Archean atmosphere and rain out. It can then accumulate in ponds on land. However, early Earth was a water world. Under the water surface of magma breaks through the oceanic crust, deposits lava, which cools and hardens quickly. 3.7 billion years ago, we see the first ever continent rise from the water, Vaalbara. Made from volcanic rock, this is a small continent, smaller than Australia, but large enough for many little warm ponds to form and accumulate hydrogen cyanide. Current scientists estimate that in these ponds, nucleotides could form. This nucleotide, adenine, is basically 5 merged hydrogen cyanides. The last major biomolecule group are the lipids, or fat. Fats are long molecules with one side attracted to water, hydrophilic, and one side repelled by water, hydrophobic. The hydrophobic side is a long chain of carbon molecules. Modern life often uses glycerol on the hydrophilic side. However, this is not strictly necessary and simpler fatty acids could suffice. This last group has also been synthesized in labs in Archaean Earth conditions, not in the atmosphere this time, but in the conditions near thermal vents. Near thermal vents, it can be 100 degrees. The thermal vents release hydrogen and carbon monoxide. Ion-containing minerals near the vents can catalyze the generation of fatty acids with long chains. This is called a Fischer-Tropsch reaction. In addition, nickel sulfide-containing minerals can catalyze carbon dioxide to monoxide to support this reaction. When these fatty acids are generated near vents, their hydrophobic tails turn towards each other to minimize contact with the water. With their hydrophilic heads turned towards the water, they form micelles. This self-assembly does not require any intervention, it is just the result of oil and water not mixing. If enough of these fatty acids stick together, they form a bilayer structure, with the water-loving heads sticking out on both sides and the water-repellent side in between. This can also lead to spherical structures called vesicles. Water inside these vesicles is isolated from the ocean. Any molecule captured inside cannot escape. These vesicles also can grow by adding additional fatty acids. And if they get large enough, they can split into two. This lays kind of a basis for reproduction. Two vesicles that touch each other could also merge, allowing the addition and accumulation of their contents, which could for example be nucleotides. Near thermal vents, there would over time be a lot of these vesicles, splitting and merging. Of course, life does not spontaneously come into existence with just these vesicles splitting and merging. What can happen, however, is for just the right nucleotide to be encapsulated. These nucleotides could form a single short RNA string. The right combination of nucleotides could form a self-replicating RNA string. In lab conditions, cross-replicating RNA has been made in 2009, where two strings of RNA would copy each other, doubling the amount of RNA strings every hour, given that enough nucleotides are available. This means that once a single self-replicating RNA string is made by chance, it can start to make copies of itself. If a copy is made and the vesicles split into two afterwards, you get two vesicles with self-replicating RNA. Replicating RNA can introduce errors for the simple cross-replicating RNA strings used in the lab, the error rate was significant, 1% per nucleotide in the string. After each replication, there are 3 possible scenarios. First, the replication is a success, with no errors. The RNA string can continue multiplying afterwards. Second, the replication happened with errors, and the new string can no longer replicate, dead end. Or 3, the replication happened with errors, but the alternation causes improved replication. Somehow, the RNA from the third scenario could overtake RNA from the first scenario. This feels like a beginning of evolution. This is called the RNA World Hypothesis, a hypothetical stage in the history of life dominated by self-replicating RNA. There is some support for this beyond the experimental results. Namely, there's a piece of RNA, most basic omnipresent biomolecule. One example is ATP, the molecular unit of energy transfer, which is basically a single monomer of RNA with 3 phosphates attached. For the energy transfer part, one of the 3 phosphates can be attached to another molecule to activate it. For example, in photosynthesis, a phosphate unit is attached to a carbohydrate called ribulose. Then, in the next step, carbon dioxide is fixed. Another example is acetyl coenzyme A, which also has a single monomer of RNA and 3 phosphates. A third example is NAD, which contains 2 monomers of RNA joined by 2 phosphates. Each of these molecules is found in every single cell of every single living organism. Another piece of support for the RNA World Hypothesis is the fact that RNA can be as enzymes, just like proteins can be. These are called ribozymes. Therefore, RNA can store information, be used as an enzyme, and be a p-struct new molecule. At this stage, we have protocells capable of replication, but they still fall short of life. There's no metabolism. Research into metabolic pathways revealed some pathways that must have been present in the very first forms of life. A prominent path is that these early life forms must be able to make their own fats by fixing carbon dioxide. This would help them enlarge their cells. Carbon dioxide is not blocked by membranes, as it's a small, unreactive molecule. Carbon dioxide from the ocean could penetrate the cell and be fixed onto a carbon chain using enzymes. This could be by ribozymes, proteins, or by a metal present inside the membrane. Many of the previous discussed reactions are catalyzed by metal-containing minerals. But these metals can also be dissolved in the water, like iron. Many chemical reactions are catalyzed by metals rather than by enzymes. The reactivity of metals can be changed by having certain organic molecules bonded to them. These molecules are called ligands. Nitrogen-containing molecules are known to change their reactivity and selectivity strongly. Both proteins and nucleotides contain nitrogen atoms. These would be the beginnings of enzymes, as certain molecule-metal combinations will improve the reactions happening. These early cells did not use photosynthesis, nor did they eat other cells. They were chemotrophs. They got their energy from thermal vents. They feasted on sulfur-containing compounds, such as pyrite, iron disulfide. They use the energy of the sulfur compounds to react phosphate with ADP to reform ATP, and this ATP can be used to initiate new reactions. The sulfur energy could also power what is known as a Wood-Ljungdahl pathway, which uses acetyl coenzyme A to fix carbon dioxide into acetate. Put all these pieces together and you see many of the necessary ingredients of life coming together. About 3.6 billion years ago, when the Earth was 1 billion years old, the last universal common ancestor, or LUCA, roamed around. It no longer uses RNA for its information storage, but uses the more stable DNA. It contains and makes carbohydrates, lipids, amino acids, and nucleotides. It fixed carbon dioxide using sulfur from thermal vents. It had no predators, and its only parasites were viruses, if they already existed. Around this point in time, there must have been a LUCA that split into two. Let's call them Brother A and Brother B. Brother A went on to become the grandfather of all bacteria, while Brother B went on to become the grandfather of all other forms of life, such as the Archaea, and much later the eukaryotes as well, which would evolve into plants, fungi, and animals.